Hello, everyone. I'm Doug Snyder with SQM Specialty Plant Nutrition North America. And with me today is Dr. Katya Hora, who is the research manager for SQM International. And she is focusing on our project that includes looking at iodine as a plant nutrient. That's what we're going to be discussing today. This is a relatively new concept to plant nutrition. And we are going to take a look at everything that we have right now to date, as well as work that's been done with iodine as far as how it pertains to being a plant nutrient is concerned. Next slide. A little bit before we get into that is about SQM. Just to give you an idea, SQM Specialty Plant Nutrition is a global company. Most of us either are agronomists or have an agricultural background. We're focused optimizing plant nutrition efficiency as well as sustainability. As you'll see, we are a global presence, and our focus is to deliver solutions that deliver results. As I stated, SQM is a supplier of plant nutrition. We are based in Chile. Santiago is our headquarters. We're also taking the material from the north of Chile, from the Atacama Desert, and from there we are shipping worldwide. As far as we are concerned in America, we are listed on the New York Stock Exchange. We have production in 9 locations and distribution in over 110 countries. You may hear us from time to time refer to caliche. Caliche is a mineral that is found in the Atacama Desert. It's a rich source of iodine, also a rich source of other products such as sulfur, potassium, nitrates, and lithium. What we do with iodine is extract from water and then leach and then evaporate, crystallize, and bring to the market either in prill form or crystalline, water-soluble crystalline form, and to bring to the global market. So with that, I'll turn it over to Katya, who will take it from here.
Thank you, Doug. In this presentation, we are going to introduce iodine to you as a nutrient for plants, and also introduce the fundamental science behind iodine as a micronutrient. This is the result of phenotypic studies, genomics studies, and proteomics studies. And after that, we're going to switch to agricultural practice. How can you apply iodine in fertigation, and what can you expect to see happening in the crop after it has sufficient iodine? So let me begin by introducing iodine. A little bit about the chemistry behind iodine. Iodine is a halogen like bromine and chlorine, and as all halogens can take different forms, iodine does the same. Elemental form is iodine, which are 2 covalently bound molecules of iodine. In natural environments, it exists as iodide. It's an anion with iodine in a formal oxidation state of -1, so 1 electron added, or iodate, which is the oxidized form of iodine with 5 electrons removed. So iodine can form covalent bonds with many organic compounds, where it will exist in its elemental form, but is covalently bound to other molecules. The most famous example of that are our own thyroid hormones. Iodine is an essential micronutrient for hormone production in the human thyroid. In the thyroid gland, a compound which is called T4 is made. This is a prohormone, and you can see with the purple dots on the molecule, there are 4 elemental molecules of iodine bound to the prohormone. This covalently bound iodine is removed by specific enzymes in our body to make the active form of our thyroid hormones. And this is why we need iodine. In general, vegetables and fruits contain iodine levels which are not sufficient to meet the daily needs of humans or animals. In most soils and substrates, there is a deficiency of available iodine for plants, and this can be due to iodine adsorbing to clay soils, or iodine being immobilized in the soils or substrate with high organic matter. Iodine can also easily leach from sandy soils, and of course in intensive agricultural soils there will be depletion of the iodine stock with crop removal. So we can say that in general there's an iodine deficiency in our diet, and this is why why supplements are required to provide enough iodine to meet the daily needs of humans and animals. There are a number of solutions to prevent iodine deficiency. The most used and most known form is to add iodine into your table salt. Also, one can take mineral supplements containing iodine, and for our farm animals, they are provided with iodine in their salt lick blocks. Now, the question that arises here, what about plants? Do plants need iodine as well as animals do? Can plants also suffer from iodine deficiency? And if yes, how can we best prevent the iodine deficiency in plants? Which brings me to the fundamental science behind iodine as a plant micronutrient. In 2021, this publication appeared. It's called Evidences for the Nutritional Role of Iodine in Plants. It is a free download paper, and in it we describe the summary of 10 years of research with results that suggest a functional involvement of iodine in plant nutrition. So, of course, all the details are in this paper, which you can find with the link above, but I will give you a short summary of what we found in this paper. The key message is that plants need iodine at 3 different levels, and that will explain its benefit as a micronutrient for plants. The first one are the visible symptoms. Can you see plants suffer from iodine deficiency? The second, the gene expression that we measured to find out that plants specifically recognize iodine in the nutrient solution and respond with changes in gene expression which are unique for the presence of iodine. So it's a reaction to iodine as a plant nutrient. And the third is iodination of proteins. We looked if plants use iodine in a way which would be similar to how humans or other organisms use iodines. Can they bind iodine in proteins? Let's go to the first part, the visible symptoms of iodine deficiency in plants. All this research was done on Arabidopsis thaliana, which is not a crop species, but it's a very useful model plant, which is used by scientists worldwide because it's very small, it's fast to grow, but most importantly, we know all the genes and most of the proteins in this plant, so you can use it to study fundamental processes in the plant. Here is how iodine deficiency in plants looks like. You can see plants on the left, and these are Arabidopsis seedlings. They're planted in a floating tray with nutrient solution below. 4 weeks after planting, you see these plants here on the left, which were cultivated in a medium which was deficient in iodine. We could not measure back the iodine in the nutrient solution of these plants, and we compared it to plants which received adequate stock of iodine, in this case 10 micromole of iodine in the nutrient solution. And what you can see in the iodine deficient plants is that they are slower to develop, they flower later, and also in the end they produce less seeds compared to the plants receiving an adequate stock of iodine. This graph shows you the quantification of that research. On the left axis is the percentage of bloomed plants, and it's plotted against the time, the number of days after planting these plants in the nutrient solution containing either very little iodine in the trough or potassium iodate at 0.2 micromole of iodine per liter or 10 micromole of iodine per liter. And what you can see that in the controlled plants, for instance, in day 9 after planting, you have approximately 40% of these plants blooming, while plants receiving either 0.2 micromole per liter or 10 micromole of iodine per liter, already 85% of these plants are blooming on day 9. And the difference between the low amount of iodine of 0.2 micromole and 10 micromole of iodine is even better visible after 6 days of planting. So apparently these plants need iodine to produce flowers and fruit. Also need iodine for a good root development. And this was studied in this system. Again, these are Arabidopsis thaliana seedlings. You see here are the little rosettes of these plants, and here's the roots going down in the medium, which is an agar medium. So it's translucent and you can see the root growth and development. On the left is an agar plate where we did not add any extra iodine, but it already has a background of iodine of 0.6 micromole of iodine in the agar. Then we increased the amount of iodine in the medium by adding either 5 or 10 or 20 or 100 micromole of iodine per liter of medium here. The 5 micromole per liter is the standard concentration which is added to systems like these if researchers use the Murshiga and Skook medium micronutrient mixes. And you can see why it is added in these mixes, because the roots grow longer, but more importantly also the lateral root development is much better. It maybe not show so clearly in this picture, but it was quantified. And when you add 10 micromole of iodine in this media, you see an even better root development, but it's also possible to provide excess of iodine, but this is about 10 times bigger than the maximal dose of 10 micromole, which we would consider to be in the optimal range in this in vitro cultivation system. And when you provide more iodine, you see the plants do not directly die, but they become a bit chlorotic and the root development is not better than the control. So this was about what you can see when you provide not enough iodine or too much of iodine. And now we will switch to what you can see in gene expression following the addition of iodine in a nutrient solution of plants. And what we found here, the conclusion is that the plants react very specifically after the perception of iodine in a nutrient solution. And we also found that this is very unique for iodine. This was done using a gene chip technology. So I will not go into the details of this. You can read these in the paper. But what we did was compare the gene expression of plants exposed to 10 micromoles of iodine in their nutrient solution, measured 48 hours after the first contact of the roots with an iodine containing nutrient solution. But we did not only expose these plants to iodine, we compared it to plants which were exposed to bromide. And what you can see here in this picture is a bar with these vertical stripes, and each stripe represents one gene. And when you look at the bars for bromide, and this is the bar for genes in the roots, and here's the bar for genes in the leaves or rosette, you see that compared to a control not receiving any halogens, there is hardly any color difference, which means that the plants did not respond to bromide in the nutrient solution. While if you expose the plants to iodide in the nutrient solution, you see a distinctive red or blue coloration. So this tells us that the plants reacted uniquely to the presence of iodide in the nutrient solution. Because it's Arabidopsis and all of these genes, so every stripe is one gene, it has a name and a number, and you can look it up in a database, and you can do an analysis to understand what it means for a plant to have sufficient iodine. The conclusions of this analysis were that the plant recognizes iodine at this micronutrient concentration in the zone. The genes that are uniquely activated with iodine show iodine's involvement in communication between cells and plant organs, but also the communication of the plant with the outside. This is called calcium-dependent signaling, and it's a new aspect in plant physiology. It's being studied for its relevance on how a plant can perceive signals from the outside world and how a plant can react with that with his own processes to meet the challenges of a changing environment. So iodine is involved in this calcium-dependent signaling, and you might compare it to the human nervous system, which is also important for us to receive signals from the outside world and being able to respond to those. What did we find next? Is that after you give iodine to the root systems with the nutrient solution, iodine is taken up and covalently bound to proteins both in the roots and in the leaves. These proteins are found to have key functions in biological processes of plants, and also what was found is that iodination of plant proteins is very similar to iodination of animal hormones. So to discover this. We used a species of iodine which was made radioactive on purpose for this experiment so that we could follow its roots from the nutrient solution into the plant and find out if there are specific proteins which it will be bound to in the plant. You can see in this picture on the right, it's a picture of proteins extracted from Arabidopsis plant, and every black band which you see here is a collection of proteins of the same size which bind this radioactive iodine. And you can see that these stripes, these black bands, also appear when you repeat the experiment in lettuce or wheat or tomato or maize, and that there are some bands which point to the existence of very similar proteins which can covalently bind iodine in plants across the plant kingdom. Now, of course, the next question was, can we identify these proteins which bind iodine, and what does that tell us about the importance of iodine for plants? We identified at least 82 plant proteins, and in this slide you see the functions of these proteins which can bind iodine for the plants. In the roots, we found 40 iodinated proteins which covalently bind iodine, and these are important for general root growth, as we saw in our Arabidopsis experiment with the roots in the vertical agar plates. Also, iodine is important in the energy metabolism of these roots, which is of course also related to root growth. 15% of these proteins is involved in anti-stress metabolism of these roots. So this corresponds with our finding of the gene expression that that iodine is actually involved in the recognition of signal to the outside world and how the plant responds to these signals. Furthermore, also some of these proteins are involved in protein production and recycling. And a number of these proteins, no one yet knows what these proteins are actually doing in these roots, but we know that they can bind iodine. Now, for the shoots, there are 42 iodinated proteins found in the plant. 86% of these are directly involved in photosynthesis, but also these proteins are involved in household functions such as protein production and recycling, cell growth, but also here some are involved in the anti-stress metabolism of the plant. Now, in summary, this work shows that plants need iodine because 48 genes in the shoots and 531 genes in the roots are uniquely regulated by iodine in a nutrient solution, and at least 82 proteins are identified where the plant binds covalently iodine on these proteins. The phenotypic studies, but also literature research, shows that iodine is needed for timely flowering and fruit production. Of course, it's involved in photosynthesis and sugar production, which means it helps the plant to increase biomass, but also to develop bigger fruits. And most importantly, iodine is involved in root growth and root function, and this process called calcium signaling, which is the nervous system of the plant. So in 2021, this work was a base for international leading plant scientists to propose a renewal of the definition of nutrients of plants. The former definition stated that a plant nutrient can be called a plant nutrient if it's essential for the plant. So if you see a plant dying, if it has a deficiency of this nutrient, but nowadays it's accepted that also nutrients which have a clear benefit for plant growth, but may not be proven to be essential for the plant, can be called beneficial plant nutrients. Now, this is a new insight for plant scientists, but actually iodine has been studied for its benefit for plant metabolism in diverse crop species already in the last century and the one even before. In a recent review of 2016, it is summarized how 30 different crops in 136 evaluated experiments show that iodine can have an impact on nitrogen metabolism, photorespiration, carbon metabolism, and the antioxidant metabolism of the plant. And below you see these crop species listed. And this corresponds exactly with the fundamental mode of action of iodine, as we found in the paper in 2021. So iodine is now known to have proven benefits for plants, and it's listed among the 19 plant nutrients for improving and protecting plant health, along with compounds like silicon or selenium or cobalt, which we know are beneficial for plant growth, even if they are not always considered to be essential. Iodine is and these other nutrients are now also listed in the standard work, Marshner's Mineral Nutrition of Plants, the latest edition. In chapter 1, it is stated that plants also take up beneficial elements such as sodium, silicon, cobalt, iodine, selenium, and aluminium, which can improve resistance to pests, diseases, abiotic stresses, as well as plant growth and crop quality. The work goes on, but now more and more people are interested to study iodine as a micronutrient. So in this low dosage between the 1 and 10 micromole of iodine per liter of nutrient solution, which is beneficial for plant growth, as is shown in this publication studying iodine and the effect on building resilience in the plants against salinity. To come back to our questions, I think I have shown you that indeed plants use iodine and plants can suffer from iodine deficiency. We have shown that iodine is directly involved in very fundamental and evolutionarily conserved processes in the plant. We have shown the presence of iodinated proteins, not only Arabidopsis, but also relevant commercial crops, different plant families, and we have shown that iodine deficiency will lead to a slower reacting plant that grows less efficiently and develops fruits less well, analogous, of course, to iodine deficiency in humans. Let's move on and see how this looks like in practice. And I will show you some examples of more than 100 growers' experiences documented by agronomists of SQM to show that administering iodine as a microelement with fertigation can actually help you prevent yield losses. Because this iodine deficient plant, which I showed you in the beginning, might be what is for the grower the normal plant, because it does not show directly as an identifiable iodine deficiency unless you measure the contents of iodine in the water, the nutrient solution, or the plant. So to wrap this up, do plants need iodine? Yes. Can plants also lose yield with iodine deficiency? Yes. In our studies to find the best way to prevent iodine deficiency in plants, we found that administering a form of iodine with fertigation that is readily available for the plant is the best way. So, how to apply iodine in fertigation? For this, we developed a product called Ultrasaline K+, which is a simple way to ensure an adequate amount of iodine in a nutrient solution. And also it's a safe application because it will prevent you from over applying iodine to any degree where it will become dangerous for the plant. So growers can easily use Ultrasaline K+ as a replacement for the potassium nitrate source to which they are accustomed. And of course, because potassium nitrate is used as a source of potassium and nitrate in fertigation programs in balanced proportion to other nutrients, this ensures that iodine is also administered in the desired dosage in a balanced proportion to these other nutrients. This product also provided a good tool to ask growers for their experience of this product. We did this in a number of demonstration trials of a very pragmatic trial design. We've asked growers if they had separated fertigation systems, so the farm practice nutrient solution with the control potassium nitrate added to the total recipe, but also a field where the plants were the same cultivar, same planting date, same crop management, but they were supplied with nutrient solution from a different fertigation system where the same composition of the nutrient solution was maintained, but here potassium nitrate plus iodine in the form of Ultrasaline K+ was added in this nutrient solution. So what we are looking for in these trials is to see the effect of iodine, which is the only differentiating factor between the left plot and the right plot and see what effect we can see in the yields of the crop. Parallel to doing the assessments on the crop, we also measured the iodine concentration in these systems. When you analyze iodine in either well water or tap water or surface water or the nutrient solutions in the drip or nutrient solution collected in the root zone, you will find that in general this is far less than the 0.2 micromole per liter, as you saw in the experiments which we done on the Arabidopsis thaliana. So it's far below the range of adequate level of iodine in a nutrient solution, which we would consider to be somewhere between 1 and 10 micromole per liter. But if you apply potassium nitrate plus iodine to this nutrient solution, so on the left you have the nutrient solution with the normal potassium nitrate, and on the right you have the nutrient solution where ultrasaline K+ was added as source of potassium nitrate. Here you see that we raised the amount of iodine in the nutrient solution to the levels which we deem to be adequate. This slide is a summary of the crop responses in these trials. Listed here are 52 of our best controlled trials. So we have a well-recorded and well-supervised total season yield in these trials. We identified the control plots as having not sufficient iodine in the nutrient solution. And what you see here is that when you put yield, which the plots received where ultrasaline, so potassium nitrate plus iodine was added, if you put this at 100%, then you see that in the control plants, actually the plants showed 9% less yield in the iodine deficient control. So you could say that on average, plants will lose 9% yield if they are not supplied with sufficient iodine. Of course, from the farmer's perspective, we turn this around, and then you have 100% yield, which you would normally expect to achieve in your crop system. Well, if you add ultrasaline, you have on average 10% benefit of adding sufficient iodine in your nutrient solution. And these trials were done on tomatoes, on peppers, on cucumbers, beans, in substrate or soil grown protected cultivation systems, but also outdoor systems like strawberry or sugarcane, banana, avocado, coffee, papaya, ginger. And also we did a series of trials in roses or lettuce on water culture. So you see that the iodine deficiency can help you to have a better yield in very different cropping systems under fertigation. So in addition to just the kilograms or pounds of more yields, farmers also comment positively about a better growth of the crop or a better quality of the produce. And you can find some of these trials described also on the SQM website. But in short, what we see is a better resilience of the plant to abiotic stresses and other climatic conditions, which can cause you to have a loss of plant growth, a loss of root growth, or a loss of fruit quality in your system. So with ultrasaline, these strawberry plants here were better able to deal with salinity in the nutrient solution. Lettuce can be harvested earlier. We prevented blossom end rot in peppers because of a higher calcium translocation. You will have less deformed fruits, a general better root condition, better root growth, better fruit weight, more antioxidants, or a more favorable balance between generative and vegetative growth as in these coffee plants. So here we move on to crop responses of iodine, and we will start with the North America. So here I hand the word back to so he can explain you some of these experiments done in North America.
Thank you, Katja. Yeah, we're going to take a look now at what iodine looks like in North America, taking a look at various cropping systems where we have explored. We are ongoing with this research, so what you're going to see is what we've done over the past couple of years, and And like I said, the more we just keep on going with this. So let's take a look. The first treatment that we're going to look at was in California with strawberry-induced salinity stress. In many areas in North America, strawberries are grown in close proximity to the ocean. If you've ever been to California, you'd realize that if you travel along Highway 101, you'll see strawberries grown within a stone's throw, if you will, of the Pacific Ocean. In Florida, strawberries are grown outside of Tampa, which is not terribly far from the Gulf of Mexico. Both of these areas have problems with salinity caused by excess sodium. And with that, you'll see decreased yields in strawberries and other types of crops. So what we did was a study that took a look at iodine and its effect on crops that are affected by salinity and that are not affected by salinity. We did this using a grower standard practice. In irrigated crops, oftentimes you'll have 20 plus applications of fertigation going through drip irrigation. This is common in these types of cropping systems. So what we looked at was products containing iodine and products that did not contain iodine. The standard that we looked at was potassium nitrate with iodine, ultrasoline K+, that you heard Katja discussed a little bit ago, or without iodine, which is ultrasol K+, no iodine. So what we were doing is what's known as spoon feed applications of the product every 2 weeks in a winterberry trial between November 3rd and March 22nd of 2022. Also noting that the native iodine content of the irrigation water was very low, but many of the waters that we use will have a little bit of iodine in them just because of the relative proximity to the ocean. Ocean water being a very big source of iodine in these close to ocean areas. Frequent applications also help to ensure plant available iodine. Iodine needs to be spoon-fed in order to work the best. So what we saw is very similar to what Katja talked about a little bit ago. Under saline situations, the addition of iodine significantly helped that crop overcome saline stress. Also, what was good to see is that in the crop that was not under saline stress, the addition of iodine also helped as well. So if you will, iodine acts as a good insurance policy to help ensure the best growth of the crop in either a saline or non-saline situation. We took a look at another study in Florida. This was an initial study looking at propagation of roots as well as fruiting and flowering structures within pots. This is a scientifically designed trial propagation trees in pots sans substrate. Let's take a look at what the results were for those. What we noted is that without iodine, you would see a root structure that many folks would say, as Katja alluded to a little earlier, this looks like a normal root system. Well, in fact, when you add iodine to the mix, you in fact see greater root growth. And of course, greater root growth leads to greater utilization of nutrients that are in the soil. So yeah, people might say those roots on the left, they look normal. Well, maybe they are. But take a look at what the iodine did for the rooting structures in the plants on the right. Normal is not, it's a new normal, if you will. And with that, for the global perspective, I'll turn back over to Katja.
Thank you, Doug. Indeed, the effect which Doug showed on better root development of orange trees when they receive sufficient iodine applied with ultrasaline K+, was also seen in these type of cultivations where lettuce is cultivated on a nutrient film technique, so only water. And in these type of systems, the plants are almost by definition iodine deficient because any iodine which they receive come only from the water, and normally agricultural water sources are very low in iodine concentration. So it's not surprising that on average between 13 and 20% of plant growth is increased in these lettuces on application of potassium nitrate with iodine. You can most pronouncedly see this effect. On young plants. So here you have lettuce brida, which was evaluated only 13 days after transplant. So this is halfway the crop cycle. And here you see that the difference of the plants receiving iodine and the iodine deficient plants are very large. In the course of the next 15 days, the plants will catch up a bit, but still a grower of lettuce on NFT is able to harvest about 2 or 3 days earlier when they apply sufficient iodine. Here's an example of turkey in a trial with 2 repetitions in the greenhouse. So we had 4 sectors in this greenhouse, and alternated was application of the nutrient solution containing the normal potassium nitrate and a nutrient solution containing potassium nitrate with iodine. And you see in the first comparison between the first 2 sectors, we had 9% more total season yield, and in the second one, 12%. So it's a repeated effect. The grower normally had his roots, which looked like these on the left part of this picture, while with the potassium nitrate, he was struck by how whiter and thicker and more branched these roots are beginning, which of course helps with the early establishments of the plants. Similar in a trial in the Ukraine, this is open fields pepper. These fields were about 10 hectares in size, and they were followed using satellite images and in NDVI measurement of the reflectance of these crops during the season. And at each date, the color and the NDVI of the plants supplied with potassium nitrate and iodine through fertigation was better, which also translated into bigger fruits and a higher yield from these plots. Again, peppers, but here in the Netherlands. These were cultivated in 2019 when the Netherlands suffered from unexceptionally high temperatures for Dutch circumstances. I know other places in the world will not blink at 30 degrees Celsius outside temperature, but for the Netherlands, this is very rare. And consequently, the growers of these pepper fruits in the greenhouses expected that their yields would drop and they would have a lower fruit size. This did not happen in growers that were using potassium nitrate with iodine at that time. So they asked us if we could investigate whether iodine might not have helped to save the viability of the pollen in the flowers of these plants, because that is important for the peppers to prevent fruit abortion and to have a good size of pepper. So we investigate this. They did this with the help of Professor Gelen and Dr. Trin at the University of Ghent in Belgium. And this group is a specialist in pollen development and survival. And they use different techniques to establish that when you take these little flowers, the flower buds of a pepper plant, and you expose the whole plant to 35 degrees Celsius for 24 hours, you will let these flowers develop again at normal temperatures, and then you look at the survival of the pollen in the anthers of these flowers. And what you find that if you have plants that were grown under normal conditions with a normal nutrient solution containing normal potassium nitrate, almost all pollen dies. 3% of viable pollen in these flowers. Well, if these plants had received a nutrient solution with iodine some weeks prior to the experiment, so that these flowers actually contained enough iodine through their meiosis in a more resilient way, there were more pollen that survived this short heat stress moment. So iodine indeed seems to protect the viability of pollen under heat stress. In retrospect, we are now better understanding results of trials which we did before the time. Here's an example of cherry tomatoes in a research center trial in Almeria in Spain, where we found through the winter period. So here the cold is not, the heat is not the stressor, but it's actually cold and a low light, which is a stress factor for these plants. Here the control tomatoes formed less uniform bunches, less nice looking, non-uniform coloration, and with smaller fruit size. And also here you see we measured the amount of antioxidants in these fruits, so carotenoids, vitamin C, and polyphenols. And when you compare the control plant, so here's the normal practice, normal Almeria Research Station practice, but you substitute the potassium nitrate with a trace of iodine, then you see much better trust development, a higher quality of fruits. These fruits were heavier, but also contain more antioxidants. So this points to an effect of iodine to build a better resilience in plants, not only under heat, but also under cold stress. An additional finding in this experiment that when you measure other nutrients in this fruit, so besides the antioxidants, we also measured potassium and magnesium and other nutrients, and these were on par between the two treatments. But when we looked at calcium, you see a distinct difference between the control plants, which is the red dotted line, where the calcium remains through October, November, December, and January at a lower level, compared to fruits which were provided with potassium nitrate and iodine in the nutrient solution. Here, the calcium in the fruits is bigger. Of course, calcium in the fruits is important to prevent calcium deficiency disorders. And to go back to the Netherlands, to the high-tech greenhouses, trial with tomato on rock wool with the recirculated nutrient solution. Here we found that after a moment of stress, and in this case it was the same heat stress as I showed you for the Dutch pepper plants, we find that with potassium nitrate and iodine, the average fruit weight is bigger compared to iodine deficient control plants. But not only was the fruit bigger, it also contained more calcium. So the iodine deficient control was measured with 32 millimoles of calcium per kilogram of dry matter fruit, while with potassium nitrate and iodine, the fruit calcium concentration increased to 40 millimoles per kilogram dry matter. And doing a shelf life storage, we found that actually this higher amount of calcium in the fruit correlated with a lower incidence of storage botrytis disease in these fruits. Similar, another calcium deficiency disorder, which often occurs in peppers, but also some varieties of tomato, is a blossom end rot. And here you see on the left capia pepper, so this pointed bell pepper type grown in Morocco on a sandy soil in a shade house. And the grower here suffered normally between 30 and 40% of loss of his fruits due to blossom end rot. While in the greenhouse where you applied potassium nitrate plus iodine in the nutrient solution, this disorder was hard to find. This translated in 10% more total yield, but more importantly also 14% of this yield, of the greater yield, was of export quality, which means a higher value for the grower. So to conclude, we went from fundamental plant science to an innovation in fertigation. Science has clearly demonstrated that iodine does indeed take part in plant metabolism. We have a relation of iodine with root growth, defense from stress, timely flowering, calcium signaling and fruit production, photosynthesis, sugar production for increased biomass and increased fruit production. Co-application of iodine with potassium nitrate in fertigation offers the most convenient and, not to forget, the safe solution to supply iodine in adequate levels for optimal plant growth. With that, we come to the questions, which I'm not sure we are taking any. All right, thank you all for your time and consideration. And should you have any question, don't hesitate to reach out to us.
Thanks, everyone. Thanks for your time. Please let us know if we can be of assistance. Thank you.